Hi, my name is Sunday Humans, and in today's video, I will be showing you the MC1R protein, also known as the melanocontin-1 receptor. And the mutation that I will be focusing on is the R67C, where the amino acid arginine at position 67 is replaced by cysteine. To begin, the normal function of the MC1R is to act as a receptor on the surface of certain cells. It is a part of the G protein coupled family, also known as the GPCRs, and in response to signals like alpha MSH triggers the production of eumelanin, a dark pigment. Me personally, I chose the MC1R as a receptor because it sits on the cell surface and receives extracellular signals and transmits those signals inside the cells to activate melanin pathways. MC1R is normally located in the cell membrane, found in melanocytes, which are specialized pigment cells in animals. You can also find this protein in many animals, including humans, mice, and other mammals. For the type of change from the mutation, the R67C mutation causes the MC1R protein to become less active. And this is because the change interferes with how the receptor folds or interacts with signaling molecules, and as a result, it can no longer activate the melanin production pathway as effectively, leading to higher pigmentation or increased skin cancer. Structural and functional impact of the mutation. The main functional group in arginine is the guanidinium group, which is positively charged. These include hydrophilic, which is polar. It is also positively charged, and arginine is also considered a large amino acid. C. Arginine interactions. Arginine can form ionic bonds with negatively charged molecules, and it is very good at forming hydrogen bonds, which help stabilize protein structure or bind to other molecules. D. Mutated amino acid side chain. Cysteine. Cysteine has a thiol group as negative SH as its main functional group. Properties include it is polar, it is neutral, and it's considered to a small to medium-sized amino acid. Interaction differences between arginine and cysteine is cysteine doesn't form ionic bonds like arginine, but instead it bridges with other cysteine residues, which affect protein folding. However, in this location of MC1R, swiping arginine for cysteine removes a key ionic bonding site, which alters the way the receptor folds are function and causes a mutation. To include, the R67C mutation changes MC1R ability to send signals inside the cell, and this reduces melanin production, especially eumelanin, leading to lighter skin or fur color. And in humans, this mutation is associated with red hair and higher risk of melanoma. In beach mice, this actually helps them blend in with sandy environments, providing camouflage. Today, I will introduce you to two versions of the same protein, MC1R, also known as the melanocontin-1 receptor. This protein plays a big role in controlling pigment production in animals and humans. And arginine, this is the wild type MC1R, and in its normal form, it sits on the cell membrane of pigment cells called melanocytes, and it acts as a receptor, kind of like a doorbell when a signaling molecule like alpha MSH comes along and presses the button. Ding, 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 melanin production coming right up. Exactly. This version tells the cells how to make eumelanin, a dark pigment that protects the skin from harmful UV radiation. Now, this is when the mutation occurs. Now, the mutation has a single change. The amino acid arginine that is positioned at 67 has now been replaced by cysteine. This mutation is called R67C. Hello, is anyone there? I think my signal's not working. That's because arginine and cysteine are very different. Arginine is big and positively charged and loves water, and it helps form ionic bonds and hydrogen bonds that stabilize the protein. Cysteine, on the other hand, is smaller, neutral, and contains sulfur. It does not form the same bonds, which means this version of this protein may fold incorrectly or fail to activate properly. And this leads to the receptor not sending the signal well or sometimes at all. And that means less melanin is made, leading to lighter skin or fur. In